Scientists have discovered a new color by shooting a laser into their eye. Did they? Let's talk about it. This is a graph of the part of the electromagnetic spectrum we can see from about 380 nanometers to about 700 nanometers. And this is a Maxwell triangle, which is a way of representing the imbalances of signal from our three types of cone cell. This is my red cone cell. If I have 700 nanometer light, way over here you can see the only cone cell that has any activity, albeit a very small amount, is the long cone cell. So I'm going to perceive this vivid red hue. If I slide it over this way, this isn't going to move because throughout this whole range, only the long cone cell is picking anything up. But as soon as I move past this point, the medium cone cell starts to get involved. And you can see the color is shifting toward green. When I reach 543 nanometers, I reach this spot, this corner that it's about to turn. If I keep going now, see my third cone cell starts to get involved right there. I start going down this ramp toward violet. So let's back up to this spot right here, 488 nanometers. This is the wavelength of the laser that the scientists shot into their eyeballs in order to try to create this new color, which they've called OLO. Even though naturally all three cone cells would be active here at 488 nanometers, the blue and red were being excluded. So only the medium cone cell was active, which meant that the imbalance could move further away from L and S and enter this uncharted space, which is where OLO exists. In the retina, at the back of the human eye, the cone cells are packed in really tight and in a random way, and they're not like labeled or color-coded or anything. And so what they had to do was individually map where these different types of cells were in each subject's retina. Once they had the map of the retina in their computer, they could use it to program their laser to aim at only the green cones. And as you can see, with those cones being packed so closely together, it's really hard to focus the laser on a medium cone and not have it spill into a neighboring long or short cone. And that is why OLO, or at least the version of OLO they were able to perceive, is somewhere around here and not all the way at the extreme of what a completely isolated M-cone response might look like.